good afternoon, and welcome to the Climate Change webinar series organized by Pire Create project. I'm Natalia Ruiz, project manager, and I want to thank you for joining us today. During this webinar, we are going to learn from our experts about paleoclimatology, what proxies can we use to get information of the past climate changes, and we will go a little bit deeper into dendrochronology and speleothem science. Today we have Ernesto Tejedor, Postdoctoral Research Associate at the Department of Atmospheric and Environmental Science at SUNY-Albany. Laia Andreu-Heils, she's a Researcher Associate Professor at the Tree Ring Lab at Columbia University. And Rebecca Orrison, graduate student also at the Department of Atmospheric and Environmental Science at SUNY-Albany. If you have questions during the webinar, please write them up at the chat and we will answer them at the end. Having made this introduction, let's start with the webinar with Ernesto Tejedor.
Hi, good afternoon, everyone, and thanks, Natalia, for the introduction, and thank you for joining. So I would like to start with a brief introduction about our Pire Create project. Our main goal here is to advance in the understanding of the paleoclimate over the Americas to the point where we can inform policy and decision making about the risks of climate change from a paleoclimatic perspective and place the future projected scenario within a broad historical context. The Pire Create project brings together 5 research institutions from the US, Brazil, and Argentina to investigate the characteristics and causes of the climate change and extremes over the last 1,000 years over North and South America, mainly by merging tree ring records and speleothem samples, and also by using climate model simulations. Another very important part of our project is the educational programs for students and outreach, including summer schools, Pire academies, where we do Become a Scientist weekend. And we have also an internships to do research in Argentina, and also a program to study 1 semester abroad in Brazil. You can learn more about this and other topics related with our project in our website. So go ahead and check it out. So when I wanted to start with this global map of the annual surface temperature anomaly, we know that the climate is changing, right? We know that because we are able to measure the atmospheric variables. In this case, you have the annual surface temperature anomaly since 1880. And you can see in the map that there are some gray areas with no data, no instrumental data. And what we can see also in this animation is that the temperature anomalies are changing. So the positive anomalies will mean warmer weather, and the negative anomalies in blue will mean colder conditions. So what we see in this map is that the world that we live in today is a much warmer planet than it was just 130 years ago. So we can measure that, we can make global means temperature, and we already have seen that with instrumental data that there is an increasing warming trend since the last 130, 150 years. And the question remains, why is that? Why do we have such a warming trend? Is that part of the natural climate variability? Is that because of the anthropogenical greenhouse gases? So we need to put that into a context. So I wanted to show you just the availability of the global instrumental data we have good quality of instrumental data for the last 70 years, more or less, but then the data becomes really scarce until the Industrial Revolution, and we really have no data beyond that period. So we need to fill this gap in the understanding of how the climate system works with something to better understand this climate, and this is what we do, the paleoclimatology. So a concept that can tell us about paleoclimatology is that it is the study of Earth's past climate history. By studying this climate variability of the past, it is possible to learn about patterns and trends of the Earth's climate system. And by inferring past climate conditions using these paleo records or proxies, we can improve our estimates of the range of natural variability in the climate system and understand how this variability has changed over the time. One way that I like to say it is like the mission of us, the paleoclimatologists, is to unlock the secrets of our past, because that will allows us the key to our future. So how do we fill this gap of climatic information? So it is necessary to distill observations from the great wealth of archives that the nature contains, and also to interpret the historical records. So within these archives, we call a specific source of information that can be linked to changes in the environment a proxy. So this is the meaning of a proxy. Then past environmental changes leave evidences in these archives, and our mission then is to interpret them and try to better understand this climate system. Then according to the location, we will find different proxy records with different sensitivities. Some proxies will tell us stories about past and temperature conditions. Other proxies will tell us more about precipitation changes. Others will have information about the past atmospheric chemical components. So here I will go just through some of them and the things that we can learn chasing the traces that they have. So for example, if we look at the marine and lake sediments, these geological archives contain information about the environment that extends back nearly 4 billion years. These layers of the sediments contains clues to what the climate was like at different timescales in the past. By looking at the types of minerals, the fossils, the chemistry, or even the size of the sediments, we can tell about the salinity, the temperature, oxygen or nutrient levels, and how past landscapes have evolved and changed. So as an example here, we can better contextualize the current warming period. We live in a geological time that we call the Holocene, which spans the last 11,700 years. We are here in the zero. And it is characterized, this Holocene age, by being a more stable climatic period. And this is key for human development, as the agriculture was developed around the world. We can see that, in fact, there was even a cooling trend, only stopped the last century with the current increase in temperatures. And these two dots here indicate the projected future increase in temperature by the end of the century. And we can then have a longer perspective. And if we look back even 400, 500 million years, we can see also that there were times where the temperature was much warmer than the temperature that we have today. But what is really important is that we can see also that the current rate of change is unprecedented for these million years. So this proxy really help us place our current climatic context in a long-term context and highlight that the rate of warming is unprecedented. And now with different proxies, we can look at and seek the causes of and attributions of such changes. For example, we can do that by looking at the ice cores. We can find ice cores mainly in Greenland and Antarctica. By extracting these cores, going with these drilling machines, the deeper we go in the ice, the more information we get back in time. And these ice cores allows us to go back in time and then to sample the snow accumulation, the air temperature, and the air chemistry from the past. High rates of snow accumulation are excellent proxies for in time resolution. We can even get annual values of information. And then these bubbles in the ice cores, they preserve actual samples of the world's ancient atmosphere when the snow befell at that past time. And this, for example, here I'm holding a 200,000 year ice core. So with these ice cores, we can estimate the past composition of the atmosphere of the last 800,000 years. And we can see, for example, in green here, the CO2 concentration of the atmosphere. And in blue, we see the temperature variability of this time period. And we see that they correlate very well. And what we also can learn from this archive is the natural range of variability of the CO2 composition of the atmosphere. And we can then put a line here. And so where are we today in this CO2 concentration? So we are here. We have as today 417 parts per million of CO2 concentration, which is unprecedented in the last 800,000 years. And what is also unprecedented is the rate of this change. So we can definitely attribute this increase to the anthropogenic greenhouse gases, and then also related with the increase in temperature variability. Another proxy record that we can use and that store valuable information about past environmental changes are the coral proxies. I don't know if you've ever been to diving or if you've seen a coral reef. These corals are colonial marine animals that form reefs in shallow subtropical waters. And the corals respond to small changes in temperature, rainfall, and water clarity in a matter of months. They respond very quickly. So it makes them a uniquely sensitive climate record. From a small coral, the scientists can put together a very detailed picture of climate in the tropics. And this is very significant because much of the Earth's weather is controlled by the conditions in the tropical oceans. I don't know if you were there, but as an example, in the previous seminar, Thomas Fabata was talked about the importance of ENSO and El Niño and La Niña patterns for the global climate. Well, if we go to the tropical Pacific Ocean and sample the coral reefs there, we can then get estimates of the past temperature variations in such an important location. And this is actually what the scientists do. They sample the reefs, the coral reefs in the tropical Pacific. And by understanding the bands in the coral cells, because they can change in thickness with changes in temperature, water clarity, and on nutrient availability. So then each band can record the season's climate, and the interpretation of the record depends on how the three factors are related: temperature, water clarity, and nutrient availability. So for example, cool water rising from the ocean floor brings extra nutrients in many areas, so the cells are often thicker when the water is cool, but this is not always true. But so the scientists have to include modern observations of the coral growth to determine what the bands are saying about this climatic change. So this is a real example of a recent reconstruction of the temperature at this location of the last millennium, of the last 1,000 years. Here, with a very high resolution, we can then understand how the changes in the temperature in the ocean has been in the past, and relate those changes to, for example, mega droughts occurring in other parts of the world, or we can find relations with volcanic eruptions. So these are very, very valuable proxies to better understand how the climate system works. And the last example I wanted to show you today, it's about historical records. Now, imagine yourself in Europe 500 years ago in the 16th century. You will most likely be a farmer, and whether you eat or not will depend on that year's harvest. So on top of that, these were very religious societies. The society was very controlled by the church. So what happened when the harvest was not doing so good, and when the crops were not growing properly? So what they will do is they will ask God for rain, because that was the mechanism that they have. This was implemented across all the Catholic Europe. So what they will do is, when there was a drought, as I told you, these were very agrarian societies and controlled by the church. According to the intensity of that drought, they will start doing different ceremonies. So if the drought was not very intense, they will only do a mass. But as long as the drought keep going and there were no rain, they will perform other ceremonies until the last level, which is a pilgrimage from one place to another. They will even change the Virgin, because that was they were really in a dangerous position, and it will eventually rain. So they will keep doing this, because in the end it will work. You know, in the end it rains. So that was working, and that was working for 600 years in Europe. According to the different ceremonies, we can then quantify that or make semi-quantitative levels. Level 1 with a small drought, level 5 with a very high drought. We can have this kind of proxies that tell us about the drought conditions in the past, and also about the hydroclimatic variability in the past. For example, here we see that 1753 has a high index. That means that that year, because this gives us monthly and annual resolution, this means that that year was having a very intense drought. And we can then relate that with other independent proxies like tree rings, and then corroborate these events. And I wanted to show you also this global map for two things. First, the different proxy availability that we have. We have in green here tree ring records. We have coral in the tropics, historical documents, glaciers, lake sediments, and these records have different resolutions. Some of these records, they record environmental changes even in the monthly resolution, like the corals. Others, they give us information about the year-to-year changes. And other proxies, they tell us information about the decadal changes. All of them are really important for getting a better understanding of our climate system, and they all span different time periods. But by combining all of them, that's when the real science starts playing out. And I wanted also to bring this picture because the paleoclimatologists believe in data sharing, and this is all open data. You will see more then with Laia. And it's very important to share the data because it allows us to find new ways of understanding these proxies, and also by combining the proxies, such as in this example that I'm showing you, which is a temperature and precipitation reconstruction of Central Europe inferred from tree-ring records, we can contextualize historical events. For example, by seeing that during the Roman expansion, there was a warmer than usual temperatures, and we can also contextualize different historical events with these climatic periods. So it's very important to put all the climatic puzzle pieces together to develop the final climatic puzzle. And with this tree-ring reconstruction, I stop my presentation and I leave Laia to tell you more about the dendrochronology.
I'm Laia from the Tree-ring Lab at Columbia University, and I will give a brief introduction to dendrochronology. That is the second part of this webinar. As Ernesto explained to you, there's like a lot of different proxies that bring different information depending on the environmental factors that imprint a signal on them. It's very important to share data, as Ernesto just said, and for that we want you to show you an example of where you can find this paleoclimate data. This is a screenshot of the NOAA webpage with different data sets of different paleoclimatological proxies are loaded. So everyone, this is publicly available, can go to this webpage and download this data. As Ernesto mentioned, it's very important to share data to be able to progress in science and not slow down process of knowledge and make the best use of resources. So if we remove all the different types of paleoclimatic data and we focus just on tree rings, we can see a map like this one with the green triangles showing the locations of the tree ring proxies. And tree rings are the paleoproxies associated with dendrochronology. In order to understand a little bit better this word that can be a little bit intimidating, we have to know that have like a Greek origin, and that dendro means tree and chronology means time. So once we know that, it's much more easy to understand that dendrochronology is the scientific method of dating rings to the exact calendar year of information. So what are tree rings? So tree rings are annual growth layers that we can see in this diagram of a cross-section of a tree. These are these circles that for sure you have seen many, many times. And why we can see these circles around the tree? So the reason for that is that at the beginning of the growing season, the tree make cells of a certain diameter, a little bit whitish, that have like this coloration. But at the end of the growing season, it grows like slower, and these cells are smaller and have like thicker cell walls with a higher content of lignin, that it's a component that gives a darker coloration. So that's the reason why visually we can see these layers that just correspond at the beginning to the whitish part, to the early wood, and the darkest part to the late wood form at the end of the growing season. What we can also see here is that the ring closer to the bark corresponds to the last year of growth, while the ring in the pith corresponds to the first year of growth. Another interesting feature in this sketch is the bark that the tree uses as a protection and isolation from the exterior for insects, but more important for climate. So insulate from cold climate and protects losing humidity, for instance. And other features are the interior part of the trunk, that it's called heartwood, that is composed by dead cells and they have like a structure function of support of the tree. And then the subwood, that it's closer to the bark and have a very important function of water transportation. And why is a very important function? Because if you remember, a tree is a living being. So we need, or the tree needs, to get water from the soil that it's coming from the rain and transport all these waters through the trunk to the canopy, to the leaf. And when this water arrives at leaf level, using CO2 and solar radiation, sunlight, it's able to do something called photosynthesis. That it's a very important function because it produces two different things. One, it's oxygen and water, that it's going again to the atmosphere through the leaf, through a process called transpiration. And then a second important thing, it's carbon sequestration. So this CO2 that it's capturing from the atmosphere is fixed on our organic tissues, different on leaf or different parts of the tree, but an important part is on tree rings, and it's fixed with cellulose and other components. So coming back to the sketch of the tree anatomy, we have this bark here, the heartwood that makes like a pillar of structure to the tree, and then the subwood that it's like bringing water from the soil to the leaf, and then we have the phloem in yellow that it's bringing the sugars from the leaf to all the cells of the tree, and it's giving also like these sugars to the cambium cell layer, that it's the only part of the tree that indeed it's active and growing. That's a little bit complicated, but it's like using these sugars for growth. So this is a little bit of wood anatomy. And when societies or scientists realize about this structure inside of the trees, so it was earlier than you can expect it. The first one describing tree rings was Theophrastus, a Greek botanist, and this is explained in the Storia Plantarum, that it's a book of general history of the plants, where he described tree rings in plants, but it was not until later on that botany accepted these findings, until the 19th century. Leonardo da Vinci in the 15th century also made these observations of these rings in the branches when he saw a saw tree, and he was able to assign these growth layers to number of years and also the thickness of the layers to the weather conditions, if it was like a dry or a wet year. So he was the first one to relate ring with variability, with precipitation variability. It was not until the 20th century when it was established the science of dendrochronology by Andreu-Heils, an American astronomer, that discovered correlations between the tree rings and the solar sunspots. How we do that? So as I said, we need to take samples from the trees, but we don't like to kill trees. So what we do is using an increment board as the one in the picture, or here you can see that, and extract a sample that is just like 5 millimeters of diameter, and we bring that sample back to the lab. Once we're in the lab, it's important to remember that our objective is to assign calendar dates to all the tree rings. And for doing that, we need to use information of which year we collect the samples. For instance, last summer it will be 2019. And we assign this calendar date to this ring that is close to the bark. Then we count backwards and make notes every 10 years with 1 dot, every 50 years 2 dots. But dendrochronology is not just counting rings, it's something else. This technique is based on something in a methodology called visual cross-dating. That it's comparing the growth patterns among different samples of different trees, as we can see here. And what we are trying to see if they share the same growth patterns. That it means if they are narrow or wider in the same years. Something that it's very useful to do is to check for pointer years that are years of extremely little growth, like in that picture 1963. If we go through samples of different trees and we find this very narrow ring in 1963, we know that we are correctly dating with the calendar year all the rings of all our samples. Our second step, it's measuring the width of all these rings. And we obtain something as these black lines that you can see in this plot that can be averaged and produce a mean chronology for that site, and also allows from a statistical validation of this visual cross-dating that we have done. Why we find this synchronicity among trees? So whatever a tree can suffer in a life, it's not representative. But this synchronicity of all the trees growing the same stand are indicating which kind of climate conditions they were exposed. So climate variations, it's making an imprint on the growth of all these trees. So when we are averaging this information and matching these growth patterns, we are getting the common information from these trees that corresponds to climate variation. Thanks to that relationship between climate and forests, we can do different types of studies. We can do ecological studies when we study the response of forests to global change, for instance. Are trees more sensitive to temperature now or to moisture? Are trees storing more carbon or less than before? Are trees transpiring more? All this is important because it affects global carbon, global hydrological cycle that can also make impacts and feedbacks to climate. We can also do a different kind of study, also using this climate and tree growth relationship that are called paleoclimate studies for reconstructing climate of the past. As Ernesto said, instrumental data only lasts for the last 100 years, or in many regions, even less. So if you have like older trees in a region, using this understanding of the relationship between growth and climate, you can estimate the climate of the past. How we do that? So we are lucky because our proxy overlaps with instrumental period. So in this plot, we can see our tree ring record in red for the 19th century, 20th century, from 1890 to 2010, and summer temperatures in black. And we can see that the variations are pretty similar, and we assess that with correlations in that case. Using this relationship, we can then estimate past climate conditions. Which kind of climate information can we get from tree rings? It will depend on which environmental factor is limiting tree growth. So I'm illustrating two examples here. On the left, you can see the boreal forest in Alaska, white spruce. And in this kind of environments, the trees are sensitive to temperature. What it means is that if we have a very warm summer, we'll have a thicker ring. That if we have a very cold summer, we'll have like a narrow ring. So in that case, tree ring with variations will correspond to variations in temperatures. On the right side, we can see a site in Mongolia, Siberian pine, growing on a lava field. So these trees are very sensitive to moisture variations. In that case, ring with variations will be informing us about changes in moisture availability. How long a tree can live? So we have like trees that can last over up to 5,000 years. This will be like the oldest almost. And another example from South America, it's like the Patagonia cypress that can last 3,600 years old. But there are more species like that, but it's not like the common thing. Most of the forests can last like, or most of the trees in a forest will have ages of 100 to 300 years, so century long. Therefore, how we can extend chronologies longer than centuries. So we can use material from dead trees, for instance. Here we see in Alaska as well a tree that is already dead. So we can take cross sections as shown in that picture from these dead trees and cores from the living trees. Thanks to that, we can make this exercise of visual cross-dating of matching patterns between the samples from the living trees, the samples from the subfossil wood. We do that, this cross-dating, matching patterns, and also we can even use wood material from archaeological structures and match these patterns. And making this puzzle back in time, we could be able to extend the chronology back in time, even if trees are not millennial long. This is an example of that have been produced in our project in Pire by some colleagues. It's a reconstruction of soil moisture for the last 1,000 years. The positive values indicate a wet period, and the negative values indicate dry periods. It's starting in 800 Common Era, and then it's finishing in recent times. What we can see is a very severe and persistent 21st century drought here. We see very negative values in the recent period. But what is interesting is that having a longer term perspective for the last 1,000 years, we are able to see four mega droughts that were produced previous to the anthropogenic era. So this means that these mega droughts were produced by a natural internal climate variability. And in this case, one hypothesis was that it will be for colder sea surface temperatures in the tropical Pacific. What we have to be aware is that if there's like a natural climate variability that can produce these drought periods, now in more recent times, we can have again like these drier periods due to the natural climate variability, but we can have superimposed anthropogenic warming, making that as water availability will be even less than expected in several places. Coming back to tree rings available across the world. As you can see in this map from NOAA, we have like a lot of tree ring records in the extratropics, but nothing or almost nothing in tropical areas. The reason for that is that in the tropics, one of the reasons for that is that in the tropics, we have an absence of winter dormancy. So in In temperate climates, trees don't grow during winter, so it's very easy for us to assign to every year a calendar date. But in the tropics, this temperature dormancy, this dormancy by low temperatures, it's not so frequent or it's not happening, and the changes are mainly driven by changes in the hydrological cycle. What makes that some of the root structures of the tree rings are more complicated, and also due to high diversity of species, some species don't even make tree rings. So it's possible to do tree ring records in the build tree ring records in the tropics. It is. Here I'm showing an example from Bolivia at 14 degrees south, where although there were very challenging wood anatomy features, here in this picture we can see an absent ring. You don't see a ring in that place, but here you can see the ring, or false rings. It seems that there's a ring here, but indeed it's like ending nowhere, so it's not a real ring. So doing the exercise that I was explaining before, the visual cross-dating, we were able to match these 4 radius from this cross-section and date all the tree rings in this sample. And how we know that we did a good job? We did a good job because we use an additional alternative method, independent method, that is radiocarbon dating. We use that during the nuclear weapon testing in 1950, there was an excess of radiocarbon in the atmosphere that produced this spike, this peak. And after 1963, there was a partial nuclear test ban. So this concentration of carbon-14 started decreasing in the atmosphere. So any organic tissue formed during these years have this imprint of radiocarbon. So you can take wood material from your rings and measure radiocarbon and see if it matches with your dendrochronological dates. And fortunately for us, we did a good job cross-dating, and for this species in Bolivia, the matching was perfect. But we have cases that it's not the case. It's very complicated. And in this case, we see also the radiocarbon curve here, and with 3 different colors, 3 different trees that we test for radiocarbon. And as you can see, it's not matching that curve. That it means that dendrochronological dates are not matching radiocarbon dates. That means that there's an issue there. It's possible to do dendrochronology in the tropics, yes. It's difficult, yes. That's also true. For this reason, because we think that it's important, part of the Pire project is invested on creating more tree rings in the tropics. And these are the places that we visited last summer for field work in Bolivia and Peru. You can see some pictures of the places that we visit in Bolivia and some samples of the material collected in Peru. Just note that the ring boundaries are not so clear as in the first picture that I show. Finally, this is a Pire product. I just would say that this is a Cedrela dorata chronology of 200 years that we have been able to validate with radiocarbon. So it's very promising that in the middle of the Ecuador, 0 degrees south, we are able to find trees with annual periodicity. This gives us hope and make that our goal of this Pire project of filling up this gap of information, it's worth it. So I think that now Rebecca will talk about Speleothems.
So hi, my name is Rebecca Orrison. I'm a PhD student at the University at Albany. I'm in Ernesto's group, and I'm going to talk a little bit about Speleothems, which is another important paleoproxy and this tropical gap that Laia mentioned that we find of paleo records can be, we're starting to fill it with tree rings, but it can also be filled to some extent by Speleothem records. So what is a Speleothem? A speleothem is a mineral deposit that can be found in a cave. So here on this slide, you see a few different examples. There are many different formations that these deposits can take. This one is called popcorn. This one is called bacon. So we can have some fun with naming them, but not all of them can be useful sources of information. The type of speleothem that we like to use is looks something like this, where you can see in the red circle there's a column, and so this is called a stalagmite, like it's holding on to the bottom with all its might. It's in usually these are layers that are built one on top of another. So usually when we're doing this research, we're going into the caves and looking for different samples, either because of their placement at some point within a cave system, but very often the samples that we're collecting are ones that have already been broken, so we can leave the cave in its natural state and we don't have to destroy parts of the cave in order to collect scientific information. So what do these look like inside? Here are a few different examples. Once the samples are collected, they're cut in half and then polished. And here you can see some of the anatomy of what a speleothem looks like on the inside. And you have different layers. I think, can you see my mouse? I hope so. So here you can see not all of the layers are very easy to look at one on top of another. You have what's called the growth axis, or the place where a layer is sort of formed around can shift. So here it's angled a little bit off to the right, and here it's angled off to the left. So doing the collection of data can be a little bit complicated. Another important difference between speleothem layers and tree layers is that not every layer indicates 1 year of growth. There could be multiple years that look like they have the same chemical composition, and so you may see differences between one layer and another, but it doesn't necessarily mean that a certain amount of time has passed. Another important difference is that because we're often collecting these samples, the outer edge of the speleothem, like the top layer, may not be from a current year of growth. And so in order to understand the age of the speleothems, we can't just look at the layers and measure the layers. And so the way that speleothems are dated and we understand how old the different layers are is by using a different radioisotope than carbon. It's using the uranium and the thorium, which is embedded in the layers. And these two different chemicals, uranium and thorium, are both radioisotopes, which means that they decay over time. And so these long gashes in the speleothem are from sampling, and we collect the material from the speleothem and send it off to a laboratory to measure how much of these different radioisotopes are within the speleothem. So that gives us a sense of how old the speleothem is and how old are these different points along the sample. So once we've done that, we can go through and measure some of the finer chemical composition of what kind of creates the structure, and I'll get into the chemical composition of speleothems a little later. But what we're seeing here in this first slide on the left is one of our colleagues from Brazil, Nico, and he's measuring with this tiny little drill to collect a sample of the layers, and he'll measure all along this axis, which is a red line that I've drawn here. So he'll take many different samples, and we know that the speleothem grows from the base up, and so you can kind of think of it as the more recent period here and the older period at the bottom. So we'll collect samples along this physical axis, which is kind of like a temporal axis. From there, the amount that's collected goes into the test tubes, and it's a really small quantity. So this quantity here can run 3 or 4 different samples, and we sample and replicate the samples, so we have verification that the results we're getting are really trustworthy. And then this test tube will go into this isotope ratio mass spectrometer, which is this big machine here, and that will spit out the isotope values, and it's really the isotopes that we're looking at. But before I get into the isotopes, I want to talk about the geographical distribution of the speleothems. So each of these stars is one different cave record that's been found from somewhere around the world, and this is part of a database called the SISL, which is from different speleothems. And so this is kind of, as Ernesto was saying, the importance of data sharing is really significant in global climate science. And so this is a database that's also open and accessible on that NOAA Paleoclimate database website. And you can find, so researchers who are interested in looking at different parts of the world can use this data. So we are seeing here that there are some records available in the tropics, particularly in the Pacific Indonesian region. And this is the result of the geology of these regions. So in the orange and the red, these are regions that contain carbonate rocks. And so it's really the dependence of the speleothem is on whatever kind of bedrock is there. So you have to have carbonate bedrocks in order to have caves that are able to be developed and have these speleothems be able to be formed. So it's really important when you're doing research that, and you're looking at paleoclimate, that depending on the region you're in, you may or may not be able to use speleothems. But luckily for us in our research within South America, there are quite a few carbonate outcroppings where these caves can form. So a little bit about the formation of a speleothem. When I think about speleothem formation, I'm thinking about following the water. So speleothems can form and encode information about the regional climate, but it all starts with the rainfall. So here you see rainfall that's coming in, and as the rain falls, it goes into the soil layer. And the soil layer is really important because it contains a lot of carbon. And so the water interacts with the carbon that's in the soil to form carbonic acid. And then the carbonic acid infiltrates down through crevices and cracks and things into the ground, into this rock layer, which is called the karst layer. And the important part of the geology here is that this karst contains calcium carbonate. And so when the carbonic acid that formed in the soil layer interacts with the calcium carbonate in the rocks, that forms calcium and bicarbonate. And then this then can infiltrate down into the cave. And that can develop into these kind of cave features that you see here in this layer. So to go a little more detail into that and what the formation process is inside of the cave, the droplets, as they're coming down through the soil layer and through the karst, and they get inside the cave, form into these drips. And each of these drips has a very high concentration of carbon dioxide because it took carbon from the soil, which is a really high concentration of carbon. And then it's in this cave environment where there's less carbon relative to the droplets. And a really important process called degassing happens, where the CO2 or the carbon dioxide that's in these droplets leaves the droplets and goes into the cave environment, leaving behind water and calcium carbonate. And so it's that calcium carbonate which then crystallizes. It's a little hard to see, but you can see within this droplet on the edge here, there's the beginning of crystallization happening where the calcium carbonate comes out of the droplet and forms into this mineral. So when I'm talking about following the water from the rain, through the soil, through the karst rock, into the cave environment, what I'm really thinking about is following the isotopes. So the isotopes are within the calcium carbonate itself in the cave. So calcium carbonate is calcium and carbon and oxygen, and the carbon and the oxygen are kind of the fundamental building blocks of the speleothem. And both carbon and oxygen have isotopes, so they have a heavy version and a light version of the same atom. So a little bit into the chemistry of the speleothems. Here we have on the left, this is oxygen 16, And on the right, there's an oxygen 18. And so these have the same number of protons, so chemically they behave the same, but the oxygen 16 only has 8 neutrons, whereas the oxygen 18 has 10 neutrons. And that difference of 2 neutrons actually gives a significant enough difference between the weight of the 2 different atoms that we can track the relationship between how many of the light isotopes exist and how many of the heavy isotopes exist in the environment. And so we express the relationship between the heavy and the light isotopes using the delta notation. So this expresses that ratio between the heavy and the light. And so if our delta notation is more negative, then we can say there are fewer heavy isotopes relative to some standard reference, which is about the ocean values, or the ratio between the heavy and light within the ocean. And if it's a positive delta value, we can say there are more heavy isotopes than within that standard reference relationship between heavy and light. So that's a little bit complicated in the abstract, but looking at an example, this is an example of the isotope hydrology. And so this is the process of water vapor as it's going. So we can start down here in the ocean, and we can see that there's relatively high delta value, which means that so that's kind of going to be our standard. But during an evaporation process, you have more of the lighter isotope that's able to evaporate relative to that heavy isotope. So the heavy one is going to prefer to stay down, but the lighter one, it can go up because it's actually because it's lighter, right? So it's kind of an intuitive way of thinking about it. And then here, as we're moving along the continent, you can imagine winds coming and blowing a cloud over a continent, and as it moves over the continent, it's going to rain. And the heavier isotope is going to prefer to rain out more than that lighter isotope. So as we move across the continent, we can see that the values within the cloud are getting darker and darker as we also move up the mountain, for example. And that's telling us that the values are going to be lower. So there's going to be more of that lighter isotope and much less of the heavier isotope left behind in the cloud. And we can see that the rainfall is going to always be a little bit higher relative to the cloud, meaning that the heavier isotopes are going to fall out first. So in a sort of cartographic form, what that might look like is this example. So this is a map of the isotope patterns, the delta patterns across South America, kind of in an average sense. And we can see a few important impacts of the regional climate on the isotope patterns here. So this is called the continental effect here, where we can see this is actually the wind patterns moving across the continent this way to the west. And we can see as we move into the continent, the values become more and more negative, which means that we're getting less and less of that heavy isotope. Another impact here is this is called the altitude effect. So the Andes Mountains are here, and we can see that as wind and clouds are moving up the mountain, they're kind of raining out, you could say, of their lighter isotopes. So the values that you're getting are more and more negative. They're raining out of the heavy isotopes. So the values are becoming more and more negative, and we're having less and less of that heavy isotope left over. So what's the importance of the isotope? They tell the story of the water cycle. They tell us where the water came from, which is called the source effect. So it helps us understand something about the regional wind patterns. It tells us where the water traveled along its journey and how it moved across the continent and or up a mountain. And it tells us something about what happened along the way, if there was any recycling of precipitation or changes in wind patterns. So happy to discuss more of that offline, or if you have questions. I know that's a lot of information. A few different examples. I'll just run through one of them. We're a little short on time. This is a paper that came out a few years ago, and this is a product of the Pire collaborations. So here on the left, you can see a time series. This is present, and here we're going back in time. And these are those delta values. So these are more positive, and here on the bottom are more negative. And you can see going back over time, there were periods where the values were pretty low, or periods where the values were higher. and fluctuation back in time. And so what this author did is they looked at one particular cave, and that's going to be the one highlighted here in red. But they also looked at a few other caves from around the region, which are these other circles, and looked at this time period here, which is known as the Little Ice Age, or period of kind of global cooling, and this period known as the medieval climate anomaly, which was a warmer period in North America. And they looked at the relationships between the values of the proxies of these Speleothem during these two different time periods. And so here they're showing that there was some relationship between this new record and this record further to the south during the medieval climate period, but relatively more positive values in the other regions. And then looking at the Little Ice Age, they saw that there was a tendency overall for the values to be more negative. And so understanding the relationships between the Speleothem isotope values at different locations and at different points in time, and relating them to one another across the continent helped to give some sense that the rainfall patterns were decreased overall during this period and increased overall in the later period, and gives us some idea of what the regional circulation patterns might have been during these two different time periods. For the interest of time, I'm going to skip over this second example, but I want to thank everybody so much for your participation and listening. And if you have any questions, I will turn it back over to Natalia.
Okay, so thank you, Ernesto, Laia, and Rebecca. We only have one question in the chat. It's, how are the coral sample? I don't know if, Ernesto, if you want to...
Yeah, I can quickly share my screen because...
Okay.
Can you see my screen? Yes.
Yeah.
So what about the coral sampling? So we are in the States. We have one of the leading experts on sampling corals. This is Professor Kim Kopp from Georgia Tech University. And here, actually the example I show you was from one of the sites that they are sampling, the Palmyra Island in the tropical Pacific. So for sampling these corals, they use these hydraulic drilling machines. They extract, I think, around 20 millimeters in diameter. They extract these cores. They don't kill the corals. You can see here another example. It's a very complicated sampling, and this is easier out in the surface, but when you have to go deep, imagine all the logistics that you have to put together to sample these measurements. So this is the kind of places that they have to go to sample. Now you all want to do coral coral record science, and this is the island of the time series that I show you, and where we can get the Palmyra Island, where we can get information about the temperature changes in the ocean of the last millennium. Here's another example of sampling these coral records. Beautiful places. They see.
Okay, thank you, Ernesto. We have another question. I don't know, Laia or Ernesto. Olivia is asking about the variation in rings of trees located in growing in a historically an urban city area versus a dense forest. If there is a variation.
Yes, so thank you, Olivia, for the question. It's very interesting indeed, because do you remember that slide that I said, we will get information of the limiting factor, the environmental limiting factor, limiting growth. So in trees, a lot of times, trees are water, or they have not living in natural conditions. So it can be very difficult sometimes getting some pure climate signal from there. And in forest, sometimes it's difficult too. because one thing that I didn't have time to explain is that the dendrochronologists should look for trees that are very sensitive to climate. And sometimes these trees are isolated trees living at the limit of the distribution, because in that cases, climate conditions or environment, it's very harsh, so you can see the imprint of climate much easier. When you have trees from a forest, a very dense forest, sometimes in addition to climate, you have like other factors as well, like forest dynamics, competence between trees. So in addition to the climate signal, you can have like these noisy signals that can also kind of damp your climate signal. So it really depends on the location of the tree and if this tree is sensitive or not to climate variations.
Thank you so much for joining us today, and hopefully you enjoy this webinar. Thank you all.